Hey, it's Mrs. G, and today I'm going to be showing you the parts of a light microscope and how to use one. Let's do it. Okay, I have a light microscope here, and first we're just going to identify each part of the microscope and what it does. This is the nose piece. It holds the objective lenses in place and it allows us to rotate between them. This is the coarse adjustment knob. Turning this knob will allow you to make large adjustments quickly to the focus of whatever you're looking at under the microscope. This fine adjustment knob underneath it makes very small adjustments to the focus for greater precision. This is the arm, which supports the body of the microscope and can be used to safely carry it without causing any damage. The base supports the microscope and keeps it steady. The stage is where you'll place your slide with whatever specimen you're observing, and the stage clips keep the slide in place so that you don't lose whatever you're trying to look at. The eyepiece, or ocular lens, is the one that you look through to observe your specimen. Most microscopes have the option of switching to a higher or lower power ocular lens for greater or smaller magnification. Each objective lens has a different magnification power to allow you to see your specimen with greater or less detail. The diaphragm controls the amount of light that's allowed to pass through the specimen. Typically, the more light, the easier it will be to observe. And finally, the light source provides that light from underneath the specimen. Some microscopes have a light bulb that you turn on and adjust the brightness. This one has a mirror that you can tilt to reflect the light from the sun through a window or from a flashlight shining on it, which is what I did here. Now we're going to practice using the microscope to observe some cells. Here I have the materials I will be using so you can follow along at home. We will be using cotton swabs to collect the sample from inside our mouths, a clean microscope slide, a clean cover slip, and some sort of dye because cells are actually clear, so you won't be able to see them under the microscope without it. I'm using bromothymol blue dye, but there are many cell staining dyes that you can use if you just research beforehand. Okay, so I know it's really hard to see because it's something clear on a white paper, but before you put your specimen on a slide, you really want to make sure to check for any debris that might be on the microscope slide already, like dust or stains. I went ahead and rinsed mine with water and just let it air dry on the paper towel before I collected my specimen. And now it is time to swab. You're going to go ahead and take your cotton swab and twirl it around on the inside of your cheek for a good 7 to 10 seconds or so. And I guess just ignore the weird faces I'm making. It's awkward to film yourself doing that. Now just twist the swab with the cells in one small area on your clean slide to transfer some of them to it. Next, you're going to want to put just one drop of whatever cell dye you're using in the same area on your slide where you rubbed that cotton swab. Now, putting the cover slip on without getting all kinds of air bubbles trapped under it is the trick. Typically letting one side fall down first so it's moving from right to left or vice versa kind of takes care of that, but you'll see there that I did get one little bubble trapped underneath it, which I'm not too worried about. If there are a lot of bubbles, you may want to pick it up and try again, but 1 or 2 doesn't bother me too much. Okay, now that we've done all the prep work, it's time to actually use the microscope. Let's position the slide over the place where the light will come through and then secure it down with the stage clips. Now we want to be sure that we're beginning with the lowest magnification objective lens, the shortest one. And this is just so that you can center on exactly the spot you want to observe close up later, because it's really hard to move around and find anything once you're on the highest magnification. I'm going to use that coarse adjustment knob to bring that lens as close to the stage as I can get it. And then I'm going to adjust the light source. Remember, it could be a light bulb or a mirror until I can see light through the ocular lens at the top. From there, I'm just going to use the fine adjustment knob to make the image as clear as I possibly can get it. And here's what I saw at that lowest magnification level. Not super clear, but you can definitely see the little dots that are my cheek cells, which is really, really cool. Once you're ready to move on to the next magnification level, you absolutely need to use that coarse adjustment knob to raise the lenses up away from the stage first. This is incredibly important because if the next lens is long enough to touch the slide, it could scrape the lens and the slide, which could damage both. Once the lenses are safely out of the way, switch to the next highest magnification and bring it down close to the slide without touching it. Here's what I saw at that middle magnification. As you can see, there's a bit more detail than the first lens was able to give us. Follow the same steps to observe your specimen on the highest magnification. Pulling the lenses away, switching to the next lens, and then pulling them back down towards the stage without touching that slide. I might have gotten a little close there. There we go. Notice the two cells toward the bottom that show much more detail than the previous two lenses could. Now you should always reset your microscope when you are done to be ready for the next experiment. That means pulling the lenses away from the stage, putting the lowest magnification back on, and then removing and cleaning your slide and cover slip. Thank you so much for watching. If you have any questions about this video, or if you are interested in private tutoring in math and science over video or in person if you live in the Kansas City area, please don't hesitate to reach out to me via email, which will be on the screen. If this video is helpful to you, I would really appreciate you subscribing to my channel. And as I always said to my students before they left my classroom, be good, be safe, and make sure you come back with all of your limbs attached correctly.